Next we come to biocatalysis. There are a large number of reactions now are conducted by using either microorganisms or enzymes isolated from microorganisms. The microorganisms themselves was in the earliest example were used for preparing the human insulin by a company called Upjohn in America. It is one of the earliest exploitation of a microorganism. The insulin is a long polypeptide. Polypeptide means amino acids reacting with each other. Several amino acids form a long chain, and this long chain amino acid, as it is present in the human insulin, was first analyzed. And the analysis la என்னென்ன peptide இருக்குன்னு கண்டுபிடிச்சாங்க. இந்த peptides எல்லாம் நார்மலி இந்த செல்குள்ளே இருக்கிற டிஎன்ஏலேருந்து மெசஞ்சர் ஆர்என்ஏன்னு ஒரு காம்பவுண்ட் ஃபார்ம். இந்த மெசஞ்சர் ஆர்என்ஏ வந்து இந்த பெப்டைட்ஸ் எல்லாம் ஒரு லைனாக அரேஞ்ச் பண்ணி இந்த பெப்டைட் பாண்ட் ஃபார்ம் பண்ணுறது. திஸ் இஸ் கால்டு ட்ரான்ஸ்ஃபர் ஆர்என்ஏ டேக்ஸ் தி அமினோ ஆசிட், gets it into messenger RNA. The messenger RNA forms a polypeptide chain. இதுக்கு வந்து they used the DNA from Escherichia coli. This is a bacterium that is present in the human gut. நம்ம சாப்பிட்ற இடத்துல இன்டெஸ்டைன்லாம் இந்த Escherichia coli இருக்குது. அந்த கோலையை ஐசோலேட் பண்ணி ஒரு ஃபெர்மெண்டேஷன் டேங்க்கில் இந்த அமினோ ஆசிட்ஸ் எல்லாம் போட்டு they reacted and they got human insulin. இதுக்கு முன்னாடி the insulin that was produced, the either bovine insulin from cattle or porcine insulin from pigs. இந்த இன்சுலின்லாம் there are some compounds which may be allergic. சில பேருக்கு இந்த இன்சுலின் ஒத்துக்காது. இந்த அப்ஜான் இந்த இதை கண்டுபிடிச்ச அப்புறம் the actual insulin that is produced is exactly the same as human insulin. ஸோ அதனால் இதில் ஒன்றும் அலர்ஜிக்கெல்லாம் சான்ஸ் இல்லை. ஸோ திஸ் வாஸ் ஒன் ஆஃப் தி ஃபர்ஸ்ட் எக்ஸாம்பிள் ஆஃப் பயோகட்டாலிசிஸ். இது வந்து லார்ஜ் பெப்டைட் செயின் இந்த மாதிரி லேபில் பண்ணுறதுனாக்க ரொம்ப கஷ்டம். வேரியஸ் சால்வன்ட்ஸ் அதெல்லாம் டாக்ஸிசிட்டி, ரீசைக்ளிங் எக்ஸெட்ரா has to be seen. Now there are other type of reactions using this. They are accomplished by using either microorganisms like previously I said Escherichia coli, or various enzymes isolated from biological sources. Some examples are hog kidney acylase. From pig kidney you isolate this enzyme acylase. Acylase means it will acetylate a particular hydroxy group in the molecule. Lipase, which will hydrolyze the fats. Lipase from Candida antarctica. Monoamine oxidase from Aspergillus niger. This Aspergillus niger is the microorganism from which monoamine oxidase is produced. Yeast is used in the manufacture of human insulin, that is Escherichia coli, and using recombinant process. This technology of producing insulin is called recombinant technology, where the messenger RNA coding for the synthesis of the polypeptide is inserted into the molecule of yeast, and the modified yeast is grown in large fermentation tanks. The insulin is isolated by suitable methods. Now biocatalysts using yeast with another type of compound. If you have this beta keto acid ether, the yeast is biocatalyst for chemoselective and enantioselective reduction of this ketone. Chemoselective means இங்கே வந்து 2 CO group இருக்கு. It will select the reaction by reducing this ketone. this is a ester group, this is the keto group. The keto group selectively reduced. Not only that, it will reduce by putting the hydrogen in the front and the hydroxyl group in the back, like I have seen. So this particular hydroxy group is produced to the extent of 94%. This is called enantiomeric excess, EE. That means this enantiomer is present 94%. The other enantiomer, where the hydroxyl group is in front, hydrogen is back, அது 6% தான் form ஆகுது. So it is a very, very efficient synthesis of the alcohol by using a biocatalyst, yeast. The hog kidney acylase it is used for deacetylating the acetyl group. இப்போ 2 amino acid இருக்கு. The amino acid is acetylated. In order to separate the amino acid, first the amino acid acetylated, and the amino acid is optically active. It has 2 different enantiomers, one R, one S. It is also designated as one D and L. D and L are mirror images of each other, but they cannot be superimposed, therefore they are optically active. In the D compound and L compound mix பண்ணி the hog kidney acylase போட்டாக்க, this is deacylated amino acid, this is L amino acid. The L acetyl amino acid is getting hydrolyzed, the acetyl group is removed. அதனால இங்கே amino group வந்துடுறது. இது you form L amino acid. இந்த இதில் amino acid group acetylated, அது அப்படியே இருக்கு, அது hydrolyze ஆகல, the deacetylated amino acid. So you can take this L amino acid and it's separated now from the 2 isomers L and D. So this is optically active. In some reactions you need L amino acid and D amino acid does not do the reaction. In the reaction use பண்ணி L amino acid isolate பண்ணணும். Resolution, in the process on the L and D separate பண்ணுற processக்கு resolution of racemic mixture. D and L together, though they are optically active, the D and L சேர்த்து வச்சாக்க ஒரு சொல்யூஷனில் they will not rotate light. The D's rotation will be compensated by L. So that is in the resolution of racemic mixtures, this method is called resolution of racemic mixtures, because D and L are the racemic mixture, அதிலிருந்து L மாத்திரம் separated. So this is called resolution, and this is reported in the journal Biochemia 1980, 45, 1361. This is another reaction where this diketone, இது active methylene group, இந்த CH2 முன்னாடியே பார்த்துருக்கோம். 2 keto குரூப்புக்கு நடுப்புற ஒரு methylene group இருந்தாக்க அது it is called active methylene group. அந்த methylene group the mutant candida group, this enzyme CALB can be used. It adds the double bond here and you get this diketone with this other ketone group present there. So this is another very important reaction where this enzyme is used to do the reaction. This is mutant candida antarctica. This is CALB, Journal of American Society, American Chemical Society, 2005, volume 127 and number page 17988. Next we go to monoamine oxidase. This is isolated from or organism called Aspergillus niger. அதிலிருந்து இந்த monoamine oxidase, suppose you have same situation like the previous one, where we have seen the resolution, you have R amine and S amine, like D and L. R and S is another nomenclature. Suppose you put it in monoamine oxidase, this goes to the corresponding imine, this gets oxidized. H and H is removed, you get double bond NH. Suppose you have borane ammonia, if you put 10 equivalents, you can reduce this. When you oxidize it, only S is oxidized, R is not oxidized. If you take this, you reduce it with borane, this will produce both R and S. And then when you do this monoamine oxidase, only S will be oxidized, for eventually this will be completely converted into S. So, this cycle allows to convert RS mixture to R amine. This R will be unreacted and this will be formed exclusively, and S will be not fully, it will be RS mixture.